In 2015, researchers working in 3 laboratories, Copenhagen, Harvard, and Leipzig's Max Planck Institute for Evolutionary Anthropology, began extracting ancient DNA from skeletal remains recovered from burial sites across Saxony-Anhalt, Germany. The bones dated to approximately 4,600 years ago. What the analysis returned was not what anyone had anticipated. The individuals buried in those graves shared virtually no meaningful genetic continuity with the Neolithic farming population that had built and occupied Central Europe for the previous 2,000 years. The farmers had not simply declined or migrated. Their genetic signature had, within what appeared to be a matter of centuries, been almost entirely displaced. The strontium isotope ratios in the teeth confirmed that many of these new individuals had originated somewhere else entirely, far to the east, out across the open steppe. This was not migration in the familiar sense. This was replacement at a scale and speed that the word migration struggles to contain. If the people we have long identified as the founding population of Germanic Europe were not descended from the farmers who cleared the forests and raised the monuments, then who were they? Where did they come from? And what happened to the civilization that was already there? The farming world that existed before this event was not primitive. It was not a thin cultural veneer stretched over an empty landscape. By around 5,500 BCE, agricultural communities descended from Anatolian migrants had established themselves across Central Europe in dense, organized networks. These were the people of the linear band pottery culture, known as LBK, and the genetic record is unambiguous about their origin. They were not the descendants of European hunter-gatherers. They were the biological children of a farming expansion that had moved steadily westward out of Anatolia over several millennia, carrying grain agriculture, domesticated animals, longhouse construction, and elaborate burial customs with them. By the time they reached what is now Germany, they had been building and farming for generations. Their burial grounds were communal, their trade networks stretched from the Aegean to the Atlantic, and their settlement patterns suggest a population that believed it had found its permanent home. Ancient DNA from LBK sites confirms that this ancestry was distinct and coherent, a recognizable genomic signature that persisted for over 1,000 years across a broad swath of the continent. This was not a fragile or scattered population. It was an established civilization in its foundational phase. Then, beginning around 2900 BCE, it started to disappear from the genetic record. Not slowly, not through the kind of gradual blending that characterizes most long-term cultural contact. The genomic signal suggests something more abrupt, a shift so pronounced that researchers at first questioned whether their sampling was skewed. It was not. When the data was confirmed across dozens of sites and replicated independently, the same pattern kept appearing. The Neolithic farmer ancestry that had defined Central Europe for millennia was giving way, at extraordinary speed, to a genetic profile originating thousands of miles to the east. That profile had a name. The Yamnaya were semi-nomadic pastoralists of the Pontic-Caspian steppe, the vast grassland corridor stretching from modern Ukraine through Russia and into Kazakhstan. Their own ancient DNA, extracted from burial mounds called Kurgans, showed a genomic composition distinct from anything previously documented in Europe, a mixture of Eastern European hunter-gatherer ancestry and a lineage tracing back to populations in the South Caucasus and northern Iran. They were not farmers. They left no known tradition of monumental architecture, no written records, no evidence of grain cultivation on any significant scale. What they did leave, in burial after burial across the steppe, were cattle bones, horse remains, wheeled vehicles, and weapons. When their genetic descendants began appearing in Central European graves around 2900 BCE, the questions that followed were not small ones. How did a population of steppe pastoralists come to dominate the genetic landscape of a continent they had not previously inhabited? And what exactly happened to the people who were already there? The first solid genomic answer came in a landmark 2015 paper published in Nature, produced jointly by David Reich's laboratory at Harvard and the Copenhagen Ancient DNA Group, analyzing skeletal remains associated with the corded ware archaeological culture, a horizon spanning from the Rhine to the Volga, dated approximately 2900 BCE to 2350 BCE, the researchers found that corded ware individuals derived roughly 75% of their ancestry from Yamnaya-related steppe populations. The number was not subtle, and the cultural markers aligned with it precisely. Where LBK communities had buried their dead collectively, corded ware burials were individual. Where LBK ceramics were painted and geometric, corded ware pottery was decorated with impressions of twisted cord. The battle axes found in corded ware graves were not farming tools. The cultural and genetic discontinuity were simultaneous, which matters because it is exactly the pattern you would expect if the newcomers had not simply traded ideas with the existing population, but had, in some meaningful sense, superseded them. The steppe ancestry signal has been replicated by multiple independent laboratories since 2015. That part of the claim is not seriously disputed. What remains contested is the mechanism, the how, and the why behind a demographic shift that left so little trace of resistance or gradual integration. A second line of evidence, independent of the DNA, deepened the problem rather than simplifying it. Strontium isotope ratios preserved in human tooth enamel record the geochemical signature of the water and food a person consumed during childhood, effectively a geographic fingerprint of where they grew up. Analysis of corded ware burial sites in Germany and Poland found a striking pattern. The females buried at these sites frequently showed strontium signatures inconsistent with local geology, suggesting they had originated in distant regions. The males were more often local. Whether this reflects long-range marriage exchange, population movement under social pressure, or something more coercive cannot be resolved from isotope data alone. But the fact that multiple measurement systems, genetics, isotope chemistry, and burial architecture were all pointing toward the same disruption made the anomaly considerably harder to set aside as a methodological artifact. The dietary signal added another layer. Nitrogen isotope analysis of bone collagen from the same sites showed that steppe-associated individuals relied significantly more on animal protein than the farming populations had. The bodies themselves encoded the cultural shift in their chemistry. But before the Yamnaya migration, there was already something wrong in Central Europe. The LBK farming world had not been collapsing peacefully. Excavations at Aspern-Schletz in Austria, ongoing since the 1980s, uncovered the skeletal remains of more than 200 individuals, showing clear evidence of perimortem trauma, wounds inflicted at or around the time of death. The demographic profile of the dead was the detail that most unsettled researchers. Women of reproductive age and young adults were significantly underrepresented, or absent entirely. In forensic terms, that pattern is not consistent with epidemic mortality, where death falls indiscriminately across age and sex groups. It is consistent with structured violence, with raids or attacks where certain demographic groups were removed rather than killed. These events predated the main steppe migration by centuries. They suggest that the farming population of Central Europe was already experiencing severe internal stress before the Yamnaya expansion arrived to compound it. Violence alone, however, cannot account for genetic displacement at continental scale. For that, a different kind of force had to be involved. In 2018, a team led by Eske Willerslev at the University of Copenhagen published results that reframed the entire question. Extracting ancient pathogen DNA from late Neolithic European skeletal remains, they identified ancestral strains of Yersinia pestis, the bacterium responsible for plague, in individuals dated to approximately 3000 to 2500 BCE. These were not the flea-vectored strains that caused the medieval Black Death. They were earlier, and probably transmitted pneumonically through respiratory contact. They lacked the specific genetic adaptation that would later make plague so explosively lethal across dense urban populations. But pneumonic plague in a population with no prior exposure and no treatment still carries catastrophic mortality potential. The temporal alignment was striking. These plague strains appeared in the genomic record precisely during the window when Neolithic population signals were collapsing and steppe ancestry was beginning to surge into Central Europe. Steppe populations, having lived in closer proximity to these pathogen lineages for longer, may have carried a degree of partial immunity that farming populations entirely lacked. The biological conditions for replacement without organized conquest were present. It is the same mechanism, the same general logic, that historians readily accept when explaining why European diseases devastated indigenous American populations after 1492, populations encountering novel pathogens against which they had no adaptive history. In that case, the dynamic is considered settled history. In the European Neolithic, the same pattern is measurable in ancient DNA, but still being debated in terms of its relative contribution to the demographic outcome. What makes this more than a story about ancient population turnover is what the steppe migrants carried with them beyond their genes. The same genomic signature that now dominates Bronze Age Central Europe correlates, with remarkable geographic precision, to the distribution of Indo-European languages across the continent. Proto-Germanic, Proto-Celtic, Proto-Baltic, and Proto-Slavic, the ancestral forms of languages spoken today by hundreds of millions of people, appear to have spread through the same demographic corridors as the Yamnaya expansion. Modern Germans speak a language whose deep ancestral structure arrived with the population event that erased much of the farming world that preceded it. The genetic and linguistic geographies align too tightly and too consistently across too many regions to be coincidental. This does not mean the correlation has resolved every interpretive dispute. Competing models still exist, and some researchers continue to argue for an earlier Anatolian origin for Indo-European. But among specialists working with ancient DNA, the steppe hypothesis has moved from a minority position to the leading model, and its reach now touches the history of language in a way that feels genuinely difficult to contain within conventional prehistory. What resists easy containment is the scale of what was lost in the transition. Modern German genomes carry, on average, roughly 45 to 50% steppe-derived ancestry, 30 to 35% Anatolian farmer ancestry, and 15 to 20% ancestry from Western European hunter-gatherers, the people who inhabited the continent before the farmers arrived from Anatolia. The hunter-gatherer signal is the oldest. These were the original inhabitants, and they were themselves substantially displaced when the Anatolian farmers arrived. Yet, they never fully disappeared. Their genetic trace persisted, reintroduced in part through admixture with steppe populations who carried some hunter-gatherer ancestry from the eastern side of the continent. 3 separate populations, each with its own language, its own material culture, its own ritual world, compressed into a single genomic layer, readable from a cheek swab. The farmers in that composite are the ones whose monuments are still standing across European landscapes, and they are also the ones whose descendants cannot be straightforwardly identified. This is where the story of the Yamnaya expansion makes contact with a much older scientific argument. In the 1950s and the 1960s, the Lithuanian-American archaeologist Marija Gimbutas proposed what she called the Kurgan hypothesis, the idea that Indo-European languages had spread westward from the Pontic-Caspian steppe via mobile pastoralist populations from the Yamnaya cultural horizon. The hypothesis was rejected by much of the academic mainstream for decades, dismissed as speculative, ideologically suspect, given the political sensitivities of European ethnic history, and insufficiently grounded in comparative linguistics. Gimbutas's model spent the better part of 50 years on the margins of serious prehistory. Then, ancient DNA research from 2015 onward produced evidence that aligned with her core geographic and demographic predictions with a precision that no one had anticipated. The reversal was not driven by ideology. It was driven by genetic sequences extracted from human teeth. A model dismissed as fringe for half a century turned out to be identifying a real population event. If that reversal happened, and it did, then the question of what else the standard model may be smoothing over, what other large-scale disruptions remain underdocumented or underappreciated, is not an unreasonable question to hold. Following the ancestry signal backward, past the Yamnaya, leads to a part of the story that is even less complete. The Yamnaya themselves were not a primary population. Their genomic composition was a mixture of 2 earlier ghost populations, Eastern European hunter-gatherers, whose archaeological record is sparse, and Caucasus hunter-gatherers, whose homeland in the south, Caucasus Mountain Zone, produced some of the earliest evidence of viticulture and sophisticated metalworking traditions in the world. That cultural complexity has never been studied in proportion to its apparent genetic influence. The Caucasus hunter-gatherer ancestry that runs through the Yamnaya, and therefore through a large fraction of modern European genomes, traces back to a region where geopolitical instability, active conflicts, and chronic underinvestment in archaeological infrastructure have left critical sites unexcavated, undated, or inaccessible to international research teams. The people who replaced the European farmers were themselves composites of earlier populations, whose full cultural world remains poorly understood. The origin story of European genetic history has a missing chapter at both ends, among the farmers who disappeared and among the steppe populations who preceded the ones who replaced them. The farmers who disappeared left something that cannot be dismissed. Across the Atlantic facade of Europe, at Carnac in Brittany, at Newgrange in Ireland, at Avebury in England, and at dozens of megalithic sites distributed across Germany itself, the monuments built by the Anatolian-descended farming population still stand. These were not casual constructions. They required the coordination of large labor forces, sophisticated astronomical knowledge embedded in their orientations, and a ritual relationship with landscape and ancestral dead that clearly mattered enormously to the people who built them. The genetic legacy of those builders was substantially erased by the steppe expansion. At several sites, there is stratigraphic evidence of corded ware period reuse, artifacts or burials from the incoming population inserted into spaces originally constructed by the farming communities. Whether that reuse represents a meaningful cultural continuity, a form of appropriation, or something more ambiguous that defies easy categorization, is a question the archaeology has not resolved. The monuments are still standing. The population that built them left no confirmed line of biological descendants in the region they shaped. The people who inherited the landscape reused the sacred spaces of a civilization they had not built and may not have understood. That is not a finished story. What remains unfinished is partly a problem of the bones themselves. The critical transition window, from 2900 to 2600 BCE in Central Europe, the period during which the demographic shift appears to have occurred most rapidly, is precisely the interval that is most poorly sampled in the ancient DNA record. Some corded ware burial sites in Germany have yielded skeletal material that has resisted extraction entirely, because temperature fluctuations and soil acidity over 5,000 years have degraded the collagen and the DNA to the point of silence. The individuals interred at the exact moment of transition, the people who might tell us with genomic precision how fast the replacement moved, whether there was sustained contact or abrupt discontinuity, whether any farmer communities survived in isolated pockets, are in some cases simply unreadable. And the ethical and institutional constraints on ancient DNA work are real. Extraction is destructive. Museum collections in Germany and across Europe are governed by preservation protocols and national regulations that limit how much material can be sampled. Some of the most important transitional individuals may never be sequenced, not because science lacks the capability, but because the bones cannot withstand the process, or because access is restricted for reasons that have nothing to do with the science. Modern Germans carry in their genomes a record assembled from 3 separate populations across 40,000 years of European history. One of those populations built monuments that outlasted its own biological continuity. One arrived from a steppe corridor whose full cultural world remains only partially documented. One survived as a ghost signal in everyone, oldest, least visible, hardest to trace. The DNA has been extracted, sequenced, and analyzed across hundreds of individuals. The broad outlines of what happened are no longer seriously disputed, but the mechanism, the precise sequence of events from approximately 2900 to 2600 BCE in Central Europe, the years when one world ended and another began, still resists a complete answer. The bones that could close that window are either too degraded to speak or have not yet been found. The transition happened. The evidence of it is measurable. The full story of how it happened and what was lost in the process remains sealed somewhere beneath the landscape it transformed.